Oh, I realized what happened last time. Okay, never mind. Yes. So I know we have a little time till technically when cross starts, so give us some time to get all logged in. I am going to have a review for the exam, unit 1 exam. I'll have one on Monday evening as well as on Wednesday. So Monday evening's review is for my Wednesday exam, but I didn't want to have them wait until you guys could take it. So I have one at like 10 o'clock at night. I didn't see that isn't fair for everybody, right? So I'll have another one on Wednesday that you guys can join. It'll be in the afternoon, like I say, like 6, 7 p.m., something like that. I'm going to post an announcement about it on Canvas, so you can look out for that. And it's probably just going to be me posting a big game. Like we did the game for the lab, we're going to do a big one for I have 50 questions for the unit 1 review, something like that. Might be even more than that. And we'll just play a game with that. I'll show you that so you guys can review that stuff and see what you know, what you need to review. Me just going over the questions over and over again at that point is probably not going to be that beneficial. You guys are kind of lucky at coming in on Thursday, even though we had a long break. It didn't feel like a break to me, but even though we had a quote unquote long break. You guys, it's still Thursday, right? And we're still facing on a weekend coming up, and I'm pretty thankful for that weekend already. And so you guys get like the lucky end of me getting angry at my Monday, Wednesday classes, which is I don't ever carry that anger into the Thursday nights. So it's always nice for you. But I'll tell you what it was about. It was mostly my hybrid group that I was having this issue with, to be clear. But they were just, I was posting these announcements about, a top hat, and I'd made one like a week prior. And then I posted another one like a week later telling like, okay, here's more detail on this since people aren't really getting it. And then I would get emails from people, asking me where they should do their, pre-lab questions, or where they should post, their in-lab, or that I can't get onto Top Hat, like how am I supposed to post my stuff and all of that. And I mean, I don't know. I don't think I have to say much, right? Because I feel like if I posted those things and people are reading their emails or checking their announcements, like, I mentioned that they should, that shouldn't be a problem. So I just remind people, and I will remind you guys, just to be fair, everybody gets a reminder, please be sure you're checking your emails and your announcements regularly. Sometimes, I'm not saying it has to be anybody in here, but it could be potentially, even in the future, if you forget. Sometimes questions that you may have about procedural stuff like that may already be in the announcement that you just haven't read yet. So check the announcement, see if there's something you missed, and if there is still a question, then of course email me. But I was getting more than one email about it, and I was just like, do we read emails anymore? And I know some people don't get their announcements in their emails. And I get that, but then you have to be sure that you're checking the announcements regularly, right? So whatever. People come in. I can't find it. It is. So yeah, I'll post the link or whatever information that we need for the review. It'll be the same Zoom link that we've always been using, by the way, but I will still post it again for convenience sake. Just a reminder that when we're going to do it. I will also on Friday be hosting another review. So it'll just be chapter 7, so it won't be a long one. It shouldn't be too bad. And I will make sure I actually click the right button this time, so that whenever you guys come to join, I'll be in the right room.
I was sitting there
when is it coming? Yes, I felt so bad because like I was kept clicking over and like if you watch the video, you see me. I'm like, nobody's on still. Like I was trying to play it off like, where is everybody? Like that's what I was really thinking. Um, no, that was my fault. That was my bad. Entirely me. If you guys haven't tried out your lockdown browser, please try to do that before the exam. It doesn't have to be today or anything, but at some point, check it out. If you haven't been on it in a while, you definitely want to be sure it's working on your laptop because you don't want to find out the day of the exam. It's just going to make you more stressed out. So check it out when you can. If you guys, other things that I'm reminding myself of as I'm looking at all this millions of things I have to do too. As you're working on your group work, you know, I know some people don't particularly care maybe for their group mates. Maybe they think the group mates aren't putting in enough effort. I don't know what the case is in this class really, but my bigger classes, this is more of an issue. So if you find that you're in a group with somebody or even more than one person that isn't pulling their weight, that's what like the grading of your group mates is for. So just be sure that you fill it out honestly and let me know that. I'm asking for some grace on the first project. So if people are like not really pulling their weight on the first project, let's be a little bit forgiving. I mean, try to work with them, of course, but like be a little bit forgiving about it. And just we'll see on the second project if they start doing the same thing when you start working on it and they're still not cooperating, then definitely shoot me an email and we can talk about it. We'll see what we have to do if we have to do anything. I had students in my class before you guys this morning who we're talking about, I say students, it was one person, but we don't need to say that, right? So who was saying that she didn't particularly care, they didn't particularly care for the group work in general, you know, and I'd said, well, there's, you know, a reason that we're doing group work. And I'm sure you guys can infer it, which is simply the fact that when we go to work in new industries or maybe in the work that you've already been working, some people haven't worked, maybe this particular individual hadn't and they don't know, but you work with people a lot of times that don't carry their weight and or that you don't like or that have different opinions than you, and you have to learn how to work with them. It's just not an option. So that's kind of what this is. I'm not meant to put you guys in, you know, these situations. I'm not telling like your roommates, hey, don't do work so they learn. Like it's not that. I would hope that everybody would work, but use it as a learning opportunity, and it might suck, and that happens. And I apologize if it does suck for you. If it continues to suck, like I said, let me know if it does. But yeah, this certain group of people happened to have an argument that was, you know, it's different when you are working with somebody because you're getting paid to deal with it. And the way that I see it is maybe that's the stakes, you know, there. If you're at a job and you're getting paid and you have to put up with these people, at least you're getting paid. But last time I checked the stakes here, I mean, you're putting several grand into this class. Don't you want to pass it? Like, and I'm not saying that I would be a dick over it, but like, come on, man. It's just not a good argument, is what I felt like. So, oh well, whatever. Let's play the game, shall we? I think we're here. We're at the time now. Yes, we are. Everybody ready? Remember, you can always join in later. Let's do our review for chapter 5. So yeah, it was not cheerful before. It's been rough. Okay, which of the following is found in a eukaryote but not in bacteria? So if you ever, for some reason, were at this point where maybe you hadn't studied as well as you wanted to and you can't remember about bacteria being this particular difference or not, I would say point at eukaryotes. And if you know what that means and what they have going on, bacteria is not in that category, obviously, if I'm having you compare them. So that's what I'm trying to say. They're not in the same category. So we're looking for something that they would not both have. All right, absolutely correct. This is the nucleus. I don't know what else to say about it. Remember, bacteria and archaea do not have a nucleus, but they are cells. So this organelle turns chemical energy into cell energy. brains. All right, so yes, it's gonna be the mitochondria. That's the powerhouse of the cell. Very proud of you guys. One more color. All right, what is the function of the endoplasmic reticulum? This is one particular function of it. Okay, so this is going to be a site for protein production. There is actually a very specific aspect of the endoplasmic reticulum that we're talking about with protein production. That's the rough endoplasmic reticulum, studded with those ribosomes that are going to start cranking out all of our protein. The smooth ER is for producing other molecules, just not protein, basically. Ready, next thing. What are the organelles that contain chlorophyll? All right, most of you guys got this one. Chlorophyll, chloroplasts, it does correlate here. So yeah, you got it. Chloroplast's gonna have the chlorophyll, and that's where photosynthesis will take place. So true or false, yeast are fungi. Excellent, this is true. It's just a type of fungi, that's the kingdom that they fall into, it's fungi. So the ones we are concerned with, of course, in the class, we're talking about diseases. The yeasts, yeah, we're talking about, Candida albicans and similar, but we also have the molds. The other fungi we don't really need to care about. All right, true or false? Fungi have chloroplasts and can perform photosynthesis. We look back on this one and say, how do fungi get their nutrients? All right, that's all right. This is every class, okay? So it's not just you guys. So we didn't talk about this necessarily directly. We talked about it sort of indirectly in the lecture. And that's because we introduced the fungi as they can get their nutrients from heterotrophs. They're heterotrophs, right? So that means, we'll get into this in more detail in chapter 9, but of course everything's in chapter 9 or something like that. But in chapter 9, we will be learning about how, these certain organisms get their energy or their carbon from certain things. So if you are a heterotroph, you are not a phototroph. That's one way we can look at it. So we're not getting our energy or our carbon from the sun and carbon dioxide. All right, so they're not that. Are there any that could be like that? Well, we talked about there are saprobes as well. They decaying matter. They help with that. And then there are the parasites. So those are the only 3 kinds of the fungi that we are concerned with. And even if we weren't concerned with them, none of them perform photosynthesis. Now you know, but that was a roundabout way to get to it. But yeah, I'm not going to be that tricky. I hope I'm not that tricky on the exam. I don't think I'm that tricky on the exam. All right, do algae help to provide oxygen to the atmosphere? Well, you guys are answering this. When you are working on your group projects for the Unit 1 group projects, the idea is when you present them, you're going to come up here and actually present them to people as groups. You'll come up here. You can have only one person doing most of the talking. That's okay, as long as you're aware that I could potentially ask people who aren't talking to explain something as well. If I think that I need to, odds are I'm probably not going to be that interested in it. So I'm just being honest, unless there's some sort of red flag, right? That's how I feel about it. So you'll come and present, and I don't expect you guys to take more than 10 minutes. In fact, if you took like 3 minutes, that would be fine. So just however long it takes to explain whatever your, review thing is, because yeah, there must be review for each other. You know that whenever you guys are coming up here for those presentations and you're hearing somebody talk about, microscopes, you're hearing talk about, bacteria or whatever it is that you're talking about, that'll help you guys review. And that's part of the review as well. So anyways, sorry. Let's go on. Yes, so the algae provide most of our atmosphere. It's actually quite a lot, about 70% of our atmosphere is microbes. So it's algae and bacteria, but yeah, so these single-celled organisms have trophozoite and cyst life stages. All right, so this is the protozoa. All of you guys got that, so I don't need to really go over that one too much. You got it. Just remind yourself in your minds what the equivalent is for the bacteria. What are helminths? I know none of it's good. All right, so this is going to be the parasitic worms, which honestly might sound a little bit like the best option here, if you had to choose. Hens. All right, what are platyhelminths? Okay, so those are flatworms. That little image was of a guy. It's a fluke, which is a type of a flatworm. So yeah, platyhelminth. These remember platy, the plat part sounds like flat to me. There's no other word that's going to be sounding like flat. So at least you can lean on that. Ready. Okay. So good job, everybody. On exam day, we will have, I think it will be on our exam day. It might be on a different day, but I'm pretty sure he's coming on exam day. It's going to be one of the representatives from SOAR, and he's going to come talk to you guys about resources that they have available for like tutors and stuff like that through SOAR that shouldn't cost you anything, you know, that's available. Just so happens that this individual was one of my better students in microbiology. So he has like firsthand experience dealing with the material. So he'll be a really good source of talking to that stuff. Yes. All right. Yes. Yeah. I don't mind if you guys borrow them now and then. It's okay. It happens. So the first thing we're going to do is go over a critical thinking question. The critical thinking question idea comes from the critical thinking questions on the NCLEX. For those of you who are going into nursing, you may know what I'm talking about. There's an entire section devoted to these types of questions. These are wordy questions that give you hints to the answer, and you need to be able to basically pick out, you know, the information that you need in order to answer the question. Now, you may or may not remember some of the material that we've gone over so far and be able to answer this question on your own. I'm not necessarily asking you to. I'm going to walk you through it. That's my point with this. Remember, so there will be questions like this on the exam, in case that wasn't hinting at that strong enough. There will be some questions like this on the exam. Not the majority, but they will be there. All right. So be prepared for that. And the reason I want to be sure and upfront about all of this is simply because I don't want you to get to it and then freeze up because there's a bunch of words. Because I know we get anxious for tests. So just remember, just read through it, take a breath, read through it, and pick out the information that sounds like key information. And I'll show you how I would start if I were somebody that wasn't sure about the information, okay? So 36-year-old man presents with diarrhea and abdominal pain. So already right here, yeah, I mean, he's 36, but we haven't talked about anything being important with age. So if we haven't talked about it, guys, it's not going to be on the test. So diarrhea and abdominal pain. So you might think, okay, well, we've talked about some things that cause that. Maybe that's information. It's something. So we'll circle it just in case, all right? He reports having eaten at a new Chinese restaurant. He ordered fried rice and said that it had been sitting out under a heat lamp all day. So it's something that's been sitting out. You can imagine, even under a heat lamp, that could brew some problems, right? So, okay, so there are a few more cases that pop up associated with the restaurant. People also having eaten the fried rice. And an epidemiologist is called to the site to investigate, what's the similarities here. He discovers, or she discovers, the epidemiologist discovers that the fried rice is contaminated with gram-positive. So now we're getting the thing. Gram-positive is something, we have endospore forming. So I just put circle endospore. And then we have that it's rod shaped. So there's 3 bits of information right there all at once. What is the most likely cause of the illness given this information and these choices? So even if you had to read through it once just to get to like what it's asking, that's okay. Go back and circle the information if you need to, or write it down on your scratch paper. I'll give you scratch paper. Anyway, so our question is basically, what is the cause of the illness? So we're looking for something that's causing the diarrhea and abdominal pain that has been sitting out for a little while. Which isn't surprising, especially we have endospore forming and stuff. You guys remember the endospores, they're hardy and they're resistant. All right, so let's start with rods. That's an easy one, okay? You guys remember the shapes that we had to concern ourselves with is really only the cocci and the rods, the bacilli, right? Okay, so our cocci are the round cells. And so if this is a coccus here and it's a round cell, we know it cannot be that. We know that that's the shape of this. We do know that bacillus is going to be rod shaped. It's not going to lie to you, the name. So we know that it's rod shaped, but what do we know about the shape of salmonella or E. coli? We have nothing, right? So that doesn't give that one away. So we can't rule them out from that alone, but at least we got rid of D. To be clear, D is Staphylococcus aureus. You might not think that that causes diarrhea or anything like that, but it's actually very strong cause of food poisoning. So like if you go and eat a food and you get sick within 30 minutes to a couple hours, it's typically the staph that's doing it. The staph will produce toxins, and the toxins that it's producing are what make you sick. Not the bacteria causing infection, but the toxins that they're producing. Huh? The food itself. So we like the bacteria, whenever they're like nice and happy on the food, are producing toxins, like just to keep other bacteria away and stuff. But then when you eat that, the toxins cause abdominal pain and stuff like that, diarrhea, all that. Typically only lasts maybe 24 hours tops. So if you get a 24-hour stomach flu, you're pretty sure it was the thing that you ate earlier in the day, it's probably staph. Very, very common. Usually it's very self-limiting as well. Because you're not causing an infection, you are, as soon as that toxin is cleared from your system, you're good. Like you're not going to carry that on or need antibiotics or anything. Yeah.
So why does salmonella last for so long?
So staph does the toxin thing, but salmonella actually infects you. So it takes a little while for salmonella to grow. Typically, salmonella is going to take a couple of days to form a true infection in your body. Same thing with E. coli, if we have the bad E. coli. And it'll take it a little while, a few days, to form in your body, and then it actually causes a true infection. It might make toxins as well, but it's actually a bigger issue, right? It's making them in your body instead of on the food that you're eating. Staph isn't going to survive your gut when you eat it, so it's really only its toxin that's making it that far. But these other 3, well, at least the other 2, A and C for sure, will cause infection.
Do the toxins like come out in the feces, or are they like sort of?
That is a good question. I think they're going to degrade. I think they degrade in the colon.
In the colon. Okay.
Yeah, it's a really good question. Nobody's ever asked that.
Sometimes we tell people when they get diarrhea like this, everyone wants to take Imodium like within 5 seconds. So
Yeah.
like sometimes that stuff
Sometimes
needs to come out.
Yeah.
Like don't stop it from.
Yeah.
So I just wondered if maybe.
With the staph, unless it's super serious, and taking Imodium, because Imodium can cause its own problems.
Right.
It's like cramping and stuff like that. As long as if you have that 24-hour bug and you're pretty sure it's just going to be the 24-hour bug, taking Imodium with that's probably not too big of a deal. If you had salmonella or E. coli, though, you've got an actual infection going on. You could be causing more problems with that. So, anyways, so we have diarrhea and abdominal pain. Everybody's usual go-to is going to be salmonella with this one because it is their common. Hey, we definitely know that causes it. Don't fall for the trick, though. All right, because we have our other clues here. Yeah, we have gram-positive. So we've already knocked out rods, right, as a clue. We've already used it up. Gram-positive, can we tell, do we know, from what we have learned so far, we have not gone over anything to tell us what's gram-positive or not. right? So that's not really helpful. So we'll go ahead and knock that one out too. And already we talked about this isn't really that helpful either. It was sitting out, and that's not surprising if it's connected with endospores. So that's probably not that helpful either. So the last thing we have is endospore forming. When we learned about endospores and endospore forming bacteria in chapter 4, there were 2 genera that are associated with forming endospores. And if you were, studying and you look back on this and you were aware of this, and it weren't just so new to you, it probably would have just jumped out at you. You wouldn't have even had to read anything or knock anybody out or whatever. Endospore forming bacteria can only be Bacillus or Clostridium, and that's it. So the name has to start with Bacillus or Clostridium. So if we, were familiar with this, at this point, we would say, okay, well, all I've got left is endospore. I'm gonna try to remember. I'm pretty sure that Salmonella and E. coli weren't the endospore producing ones. It wasn't that. I remember that maybe. It's gotta be Bacillus. You could even roll it out that way if you didn't remember it directly. I would hope that because all you have to remember is 2 for endospores is Bacillus and Clostridium, and I will say them a lot throughout the course. Which doesn't help me now because we're in the beginning of it, but that's okay. That you would see endospore and think it has to be Bacillus. It's either Bacillus or Clostridium, and Bacillus is the only thing up there. So that's how we get to our answer on this. It's just having to know that. So we have all these words. It's just a load of BS, really, because really the only thing you needed to know is get to that endospore, and that immediately got you your answer. So this is exactly what I'm talking about with these critical thinking questions. Don't get caught up in the words. The words are there to give you information, but I know that when I say diarrhea and abdominal pain, most people are going to think salmonella and just pick it without getting through the other information. But remember that. Read through everything. Take a minute and really read through everything. Yeah, probably when you take the exams, you're going to need to tell yourself to take a breath. It's okay. It's okay. If you guys get to something, a question, like if I had said that the man had experienced emesis or emesis, depending on how you want to pronounce it, right? So an emetic, like if you have somebody, and I hand you an emetic bag, when my husband works at OBI, he's a supervisor for like the mobiles. So he goes out to places and they draw blood and people throw up all the time, right? So they have these emesis bags. And then you can also take drugs that are like antiemetics. That's both the same thing, okay? So if I had said up there though, that it was, an emetic bacterium or something like that causing vomiting, but I wouldn't have explained that. If you have a word like that I haven't explained in class and you're not sure what it means, ask. The worst that can happen is I'll tell you, so you're supposed to know that term, and I might try to give you hints around it if you're supposed to know it. But if you are pretty sure that we didn't talk about it, it's just a term that you just don't know what the word means, ask. I'm not trying to trip you up, okay? I don't always think about it. So let's get into the viruses. This is going to be, they are non-cellular. Remember how we had drawn on the board microbes. And our first thing that we did was cellular and acellular or non-cellular. So the viruses are going in the non-cellular category, but they infect any kind of cell ever that we've found so far. There's viruses that infect those cells. So what are they? Are they alive? What role do they play in evolution? That's a weird question to ask, but it's not as weird as you think. Their characteristics, and if they're so small, how do they cause so much damage? And what are connections between viruses and cancer? I'm sure at first it might sound weird, but then if you think about something like HPV that causes cervical cancer, then it doesn't sound so weird anymore, right? All right, so let's get into it. We usually typically are going to call viruses active or inactive. If we're scientists and we're playing this correctly and you know better, it's active or inactive, not living or dead, simply because they weren't really truly ever alive. And the way that we've defined that, remember, is based on the cell. A cell is a membrane-bound item. I don't know what else to call it. It's a cell. It's a membrane-bound structure, right, that has everything that it needs to divide and multiply on its own, to make a copy of itself. It can do that on its own. Viruses cannot do that on their own. They have to be in your cells and stealing your stuff from your cells to make that happen for them. We'll talk about the basics of that as we get through this, because a lot of times viruses are the most vague concept for people. They just think of them as being different bacteria, and they're nothing like it.
We can't really ever get rid of them. Like things that are viruses, we can just treat, but they're always there and dormant and waiting.
That is a very, okay, so astute statement, okay? Some of them. You're right with some of them, okay? Yes, so we'll get into that too. And that's part of what we're talking about with the cancer causing ones. Yeah. Because I mean, how do you cause cancer in somebody if you're just a normal virus and just doing normal virus things? These viruses are tricky and they're nasty. So we're going to talk about it. But cells, remember, divide on their own, multiply on their own. They have everything they need on their own. Viruses do not. It's actually super basic. So we can call these guys obligate intracellular parasites. Here's a word to be familiar with. Obligate intracellular parasites. The word that we can break down as well. Obligate, like you're obligated to do something. You have to do it. You're obligated to take your sister to school, or you're obligated to go to work the next morning. You're obligated. So they're obligate, so they have to do the thing. Intracellular means inside of the cell. And then parasites, we've learned a little bit about parasites. They're taking something from the host, causing damage or stealing from the host for their benefit, while the host isn't doing so hot for it. So that's a parasite. So obligate intracellular parasites. We can break that down so it's not as overwhelming. All right. As it turns out, as we were kind of saying already, between 40 to 80% of the human genome seems to be remnants of ancient viral infections. And I don't mean like you had the flu earlier this year. I'm talking about like our ancestors way back in the day got infected with viruses that became part of their genome, and now we carry that information with us. They don't typically code for anything or do anything that we know of, typically, but they're just there. It's just extraneous nucleic acid in our genomes. It's pretty crazy to think about that much of your genome being trash, basically, of old virus stuff. We do consider viruses to be part of our normal microbiome because they can't really be out on their own. They're not living outside of the cells, chilling on our skin or something. They have to be infecting the bacteria, or they have to be infecting our cells. So they have to be doing something like that. This is a good review slide. I'm not going to read through the whole thing right now, but it kind of summarizes everything we're about to go through. So it's a good one to come back to remind yourself of it. This is a size comparison chart showing us yeast cells, which our cells are actually larger even than yeast cells. So this is a yeast cell. This bottom part of it is the full size, and the top part is like a budding new cell coming off of it. This is E. coli. This is strep. These two are the largest viruses that we know of in existence, Pandora virus and Mimi virus. This is a bacteria called Rickettsia. Rickettsia rickettsii is the cause of Rocky Mountain spotted fever. So it's just bacteria though. All right, and then I'm sure you can guess we're going through the viruses that cause disease. Because these guys, these bigger ones over here, these guys I don't know what they do. They're just big, okay? I don't know what they do. I'm sure they have purpose, just I don't know what. All right, not diseases for humans. We have here herpes, all right, rabies, HIV, flu, adenovirus, which they use as a like shell vector for one of the types of the COVID vaccines. I can't remember which ones are which anymore. I don't know which ones, but that's why you can't mix them up. That's why you have to get the same ones every time, because some of them are, they're going to give you info on COVID inside of a shell of an adenovirus. They put it into your body, and your body starts acting like it's a virus, starts making COVID proteins. And then your body responds to the COVID proteins that are going to be on this adenovirus. So that's one way to get your body to respond to COVID proteins. Another way to get your body to respond to COVID proteins is by taking vesicles that you can make, sort of just like man-made lipid bilayers, and just putting some mRNA in it and shoving it into your arm, and your cells will take up that mRNA because it's got that lipid cover onto it, so it'll sort of merge with your cells. You'll take up the mRNA, and then your body's going to do what it does with mRNA, make protein. One of the proteins that it's going to make is the COVID spike protein, and put that on the surface of your cells. And then your body will react to that because it's foreign. And now you've got antibodies to COVID that are built up in your body in one of the weirdest ways possible. And it's super weird. In fact, the person who developed that won the Nobel Prize for it. And as luck would have it, aren't you all so blessed that I took my immunology in graduate school with that man? So I get to study with this man that ended up getting a Nobel Prize. Not that I'm ever going to achieve that or anything, but I feel like kind of, you know, I don't want to say special, but it's kind of cool to get to say that, you know, but yeah. Anyways, so adenovirus is part of that whole play. Not his part, but it is important. T2 bacteriophage. Normally adenovirus, by the way, it causes colds. It's just a cold virus. One of the 200 different kinds of cold virus that are known. Bacteriophage is a virus that infects bacteria. So we call them bacteriophages. So that's that one. This is polio. This is yellow fever virus, this little guy right here. And then down here, this is the one I wanted to get to. This molecule here is just hemoglobin, that little dot there. Just hemoglobin, not cells, not the red blood cells, just hemoglobin protein. Look at the size of hemoglobin compared to yellow fever virus. So when I'm telling you guys that these are small, I mean, they are basic, and they're small, but they cause massive disease. If you don't know, yellow fever is a pretty serious disease. You can die from it. We don't have it here in the United States, but they have them in the tropics. You can get vaccines for it. But yeah, look how tiny it can be. So we're going to move on. These guys don't bear any resemblance to cells that we know of. They lack any protein synthesizing machinery, and they only have the parts that they need in order to invade and control a host cell. So we're going to talk about the basic parts of viruses. So as we look at this, we have the basic parts of a virus. First, I guess the most important thing is going to be the nucleic acid genome. Like as a genome, it has to have a genome, means the code for whatever. So this is going to be its nucleic, just like us, we have nucleic acids too. And that makes sense. It would want to have a similar kind of nucleic acid to what we have, so that we can use our machinery. Otherwise, you know, what's the point? This is our genome. And then the other part that you absolutely have to have, you only need two. This is our bare minimum though. Okay. That's the capsid. The capsid. Only need a nucleic acid genome and a capsid. That's our bare minimum. What's the capsid? It's a protein coding, protective protein coding. And what is it protecting? The, you know, the only other part of this that's going on. There's literally only going to be two parts so far. So that's what it is. And it's made of subunits. And those subunits are called capsomeres. So those come together to assemble and make the capsid. Just right now, nucleic acid genome has protein around it to protect it. That's all you need. Some viruses, that's all they have. That yellow fever virus, the adenovirus, the what was the other one? Forget what it was. Anyways, those guys, they don't, that's all they need. Poliovirus, that's the other one. They just are this. And so what genes do they need in their genome? They need genes for capsomeres. That's it. That's all the genes they need. And so it's typically going to be repeating into the same capsomeres that are going to come together and spontaneously assemble to create the capsid. And they'll do that on their own just because chemistry. You know, we talked about the hydrophobic and the hydrophilic, how they want to get together and whatever. That dictates how the capsomeres are going to come together and work together. So that's all you need, really. But now we can get a little bit more complicated with some of the more complex viruses. Let's see. Let's go ahead and get into the next part, which is going to be the, well, I'll say this next thing first, okay? Kind of a separate part. Spikes. Now, I know you guys have seen pictures of the spikes on COVID. We saw them kind of a little bit on that adenovirus. They're literally just coming off the side. This has little spikes coming up, these little pink guys that are coming up. That's the spikes. And what's a spike? Spike is a glyco. You guys remember what this means? Sugar, right? Protein on the surface, like a spikes on the surface of it, these glycoproteins allow entry into the host cell. So I like to refer to them as like a lock and key mechanism. So glycoprotein, we'll say it's like a key. This glycoprotein spike, these spikes on the outside of the viruses, will come along and interact with, molecules on the surface of your cells, right? Sometimes it's very specific. Sometimes one virus's spikes will interact with specific molecules only found on human liver cells, and that's the only thing that they can infect. Other ones have spikes that will be found on all, let's say, all mammal cells. Like it's some sort of, their spike interacts with some sort of protein that's found on every single mammal cell ever. And so that's the door that it's looking for. That's the lock that it's looking for there, right? So these guys will only interact with whatever the lock is that they're looking for, whether it is only on human liver cells or it's on all mammal cells. Okay, so we'll talk about this in a little bit, but as you guys get to hear my lecture about my favorite disease of all time, and I love scaring people half to death with it, and it's gonna be good. So so there's that. Oh yeah, the other thing I was gonna try to do. So you see this one has a circle around it. It looks like out on the circle. That's different than what I'm telling you a little bit. So this part of it is called the envelope. And it's not unlike the cell envelopes of bacteria. So anytime you guys hear envelope, we're talking about the entire covering of something. This doesn't include the capsid here because the capsid is a different part of it. So the envelope is that, it's going to be a lipid bilayer. I'm just going to give away the secrets here, okay? It's going to be a lipid bilayer, just like we have the lipid bilayer on our cells, just like the bacteria have the lipid bilayer on their cytoplasmic membrane and then the wall. And then if you're gram-negative, another lipid bilayer, right? So that whole thing, those three layers, would be the envelope for the gram-negative cells. For our viruses, they also have a lipid bilayer, but that one thing will be their envelope. It doesn't really get any more complicated than that, thankfully. But if you are one of these guys, that's just going to be these two things. Like I said, polio, for example, right? This is called a nucleocapsid, no matter what kind of virus you are. It's a nucleocapsid, we're just putting them together. And that structure is a thing on its own. You can be a virus just on its own with just that. That's okay too. It can be. Or you can take your nucleocapsid and put an envelope around it. So those are options, basically, is what I'm saying. If you're going to be just this nucleocapsid, obviously, if you are going to be only that, you need to have your spikes on the nucleocapsid, right? It's going to be right on there. So it's part of the capsid. So the spikes will be on the nucleocapsid. This is only in certain cases where it's only the nucleocapsid, right? If you are only nucleocapsid, therefore have your spikes on the nucleocapsid, then you have a special name for yourself. It's nothing tricky. Naked. These viruses are just like this. It's how they are. It's not like we took their envelope off of them. That's just how they are in nature, okay? Like the adenovirus, like polio, whatever it is, it's a nucleocapsid with spikes on the outside. If there's no envelope, it's naked. Okay? The envelope, the way that it works, is we'll have that nucleocapsid. It's going to be surrounded by, enveloped by a lipid bilayer. So let's put bilayer. The membrane is fine. Also, let's do that membrane. This is your situation. If you're an enveloped virus, this is the enveloped virus. The envelope itself is going to be that membrane. Just want to be clear, we don't. Enveloped viruses have the nucleocapsid surrounded by the membrane. If you're going to do that and you have your whole nucleocapsid inside, I'm sure you've seen that this is kind of how they're going to be. And then you have your capsid surrounding your nucleic acid. So that's your nucleocapsid. This is your envelope that we go around it. So how are you going to get into your cells? Target cells, if your spikes are on the nucleocapsid, it won't work, right? That's why we have it like that in that picture. You'll have your spikes coming off of the envelope. Viruses are pretty nasty. They're gonna come in there with their genes and, like I said, release their little genome and have your cells to start doing whatever the genome will ask it to do. Because our cells don't know better. You know, they're just reading whatever the genetic information is that's there. And so it'll go in there and have you start to making the spike proteins, the glycoproteins. You're going to do that. Your cells are very diligent and they will do that and it will stud their entire surface with spikes from this virus. And that's all it's going to do. It will knock off its own proteins. It won't be able to do what it normally would do based on those outer proteins, including sticking to other cells, for example. So completely spikes. So that when this guy's made, the nucleic acid, if it's trying to get a membrane around it, that envelope, it will literally just push its way through that envelope. It's like a blanket, like I was walking through a blanket and I just tied it around myself like that. They're just going to bud through or push their way through that cell membrane. And that's how it gets the spikes on the outside. You already put them there. So now it's walking away with spikes on its outside of its membrane at cost to your cells, right? That's how it works. I mean, it is as nasty as making it sound. Tell me that's not crazy, that these are just simple, basic machinery, basically. So yeah, some of them have a couple enzymes or something that they'll bring with them. We'll get into that later, but I don't want to get into it now. We already talked about the capsid. We talked about the envelope. That's the membrane that it can have. It doesn't always. You're naked if you don't have it. The spikes themselves are found on the surface of any kind of virus that allow it to dock with its host cells, its target, right? So here's looking at a picture of a virion that is naked, and the virion that is enveloped. And virion is just a fully formed virus, just like a singular way to determine what you're talking about. So if you're just talking about one viral particle, it's called a virion. Yeah, we talked about the capsomeres. Yeah, there's a couple different kinds of capsids that you can form. I've been drawing the ones that look like it's got, you know, triangles that are building it up. We've seen the picture of it in this other image. Look at those triangles, right? Even this one here is triangle based. It's not as clearly drawn out for us there. Those are called icosahedral capsids. So they are the ones that we're probably most commonly used to seeing. You can also have helical capsids. And the way that the helical capsids work is you'll take the nucleic acid of whatever your virus is, and then put your capsomeres, those repeating units, and just cycle them around and around in a big old spiral around your nucleic acid until it's completely covered. That can be enough. Like I said, you can be a naked virus like that. Like it says up here, tobacco mosaic virus, if you care about plants, is a virus that can infect plant cells, and it's like that. Okay, it doesn't have an envelope or anything. It's just big capsid that is helical around that nucleic acid. Most of the ones that we would be used to seeing though are going to be enveloped. So when you have a helical capsid, it's like you're making this big old, let's draw it. Here is your nucleic acids, whatever, and you're building your helical capsid around it, right? You're just going circle, 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 all the way down. So now we have our nucleic acid inside of our capsid. It's our nucleocapsid. Anyways, when you get it long enough, when you've got the whole thing, you can like just spiral it up. Imagine you've got your nucleic acid in there though, right? And then that can be surrounded by the envelope. Okay, so it can just wad itself up and then be surrounded by an envelope. So that's one way it can do it. Most of the time we're talking about the icosahedral ones. But these do exist. I just want to be clear. Yeah, like I said, normal, what we're used to saying in animal viruses, even though at this point you're probably looking at any of these and thinking this is not normal. And that's a reasonable explanation. Now let me show you not normal, guys. So this is the bacteriophage. This is a T4 bacteriophage specifically. It's like the bread and butter of all bacteriophages if you cared about a bacteriophage. But it's what it looks like legit. This is the drawing of it. This is an electron micrograph. This is an actual picture of them. So that's what they look like. And we've actually got it down to how they actually operate with this weird shape. But these are bacteriophages. We're going to get into them a little bit more in a little while. But these are viruses that infect bacteria. That's what a bacteriophage is. Viruses that infect bacteria. Cool, some more pictures. We talked about the envelope. Yes, we talked about the spikes on the envelope. All right, so, no, I don't care about this really. Okay, they can have genes. They do have genes, obviously, but some of them have a lot more genes than others. You look at hepatitis B, it only has 4 genes, whereas Pandora viruses have 2,500 genes. So it just depends on the virus, man, as far as like how many genes it's carrying around. Just to be clear, you have 20,000. So that's pretty impressive for a virus. All right, let's talk about the nucleic acids. This part is boring and horrible. So it's hard. This part's hard if you haven't taken the genetics chapter, which is why the genetics chapter comes first in the book. But I feel like we should be doing like bacteria and eukaryotes and viruses all together. I don't know. So we get to as much as we have to get. But let's talk about the genome. So their genomes can be a bunch of different things. They can either be DNA or they can be RNA. This would be like, for example, like our DNA, double-stranded DNA, the double helix. Okay. The RNA can come like what we would expect for it to come. RNA, you can just, it's always going to be single-stranded, by the way, for that. It can have double-stranded, not say it's always single-stranded. It can have double-stranded RNA, which is weird. This is something that your body will automatically see as foreign and attack, because it should not ever have double-stranded RNA. Our body sees that as strange. So that's one thing we can pick off for our immune system. It's looking out for us there, our innate immunity. Anyways, then we have the regular RNA. Our messenger RNA, which we'll learn about in chapter 6, will be single strand. It's like ready to go, ready to read, ready to match up our corresponding tRNAs with it as we're going to make our proteins and everything like that. So it's single stranded. So it could be single stranded, but it's not as simple as this. Why would it be? So it can be single stranded RNA that is positive sense, or single stranded RNA that is negative sense. So what do I mean by positive and negative sense? Positive sense RNA is like your mRNA. Your ribosomes can latch right on and start shooting out a protein. They can just read it. Negative sense RNA, you have to make the complement. Essentially, the other side of the double strand that we didn't copy when we made our mRNA. So the complement has to be made of that before we can make a protein from it. That's what that means. For now, that's all we need to know. Okay. You can also have positive sense single strand DNA or negative sense single stranded DNA. So these guys are not playing around. We can only have this, and then the viruses, depending on what virus you are, to have any of these, any combination of these. Pretty much, you can dream it up. They can have it in the sense of double strand, or in the sense of DNA versus RNA. Okay. I know. All right, let's see. So some of them, like I said, can also carry enzymes with them. You guys remember, because I just talked a little bit about it, you guys remember how I talk about, you have your DNA and your genes in your Smithsonian institution, right? It has everything of your genes in that nucleus. And that will make messenger RNA copy. That happens in the nucleus because that's where the DNA is. And then that's going to get transported out of the cell and then bind up with some ribosome, whatever, and then get read by the ribosome and get out of that protein. This is the watered down version. But DNA to RNA, that's transcription, we're just rewriting it, but still nucleic acid. The the translation, now we're turning nucleic acid into a completely different language, but that's what we call it. So this was the central dogma of biology for a long time. DNA always goes into RNA, always goes into protein. And that was the rule that you had to follow. You couldn't go back. So not a thing. Okay? Nope. Not until we met HIV. HIV broke all the rules. It's one of the greatest discoveries probably in the era, if you will, right? So first of all, someone can take with them polymerases just to make copies of their genome if they want. They'll just pack it in with themselves. Or they can pack in with themselves replicases, or they can just code for replicases that just make RNA into RNA. So we don't need the DNA intermediate at all. They can also make reverse transcriptase. This is the big daddy. Okay, if you learn anything about these enzymes that these viruses can have, it needs to be reverse transcriptase. That is what allows us to go from RNA into DNA. So we can now make DNA from that measly old RNA that we had. We got this from HIV. HIV can do that. It has an RNA genome, and it's turning its RNA genome into DNA. Now, why would it want to do that? Well, as it turns out, nobody cares about this next slide, by the way. This is just classifying. We'll get to the questions in a second. As it turns out, HIV wants to put its genes into your genes, and it does so quite successfully. It takes its little DNA copy that it made of its RNA and shoves that DNA copy into your genes, and it stays there forever. How do you get rid of something like that? Yeah. How do you get rid of genes that got put into your genes? Like, can you control that really? Of course not, right? Because that is just not right now, right? Not with the technology that we have now. Maybe we can solve that problem with CRISPR or something like that later on, but it's not as simple as just like injecting somebody with CRISPR. You guys know about that. It's used to cut DNA pretty much anywhere that they want it to. But you can just inject somebody with that, right? Or be cutting willy-nilly. How do you get it to cut out specifically what you want? Anyways, you can't right now. So when you have HIV, you have it for life. Even if you take your medications and you have a 0 viral load in your body, you still have HIV. Because at any point, if you stop taking that medication, that stops your body from making the virus for the virus. If you stop taking that, then your body's going to make more virus. So that's a problem, and it's very serious, and it's something that, of course, science is looking into right now. But it's not the only virus that does this sort of thing. If you ever think to yourself of any viruses that you or anybody that you know may have, that you get it once, and then it can come back at any time. Anybody think of anything like that? Yep, herpes is a perfect example of that. We have chickenpox. What did you say? Yes, so that's, yeah, herpes virus. So herpes simplex 1 or 2, right? Oral or typically mostly oral or the genital for 2. Herpes viruses are a perfect example of this. They insert their DNA into your genome, and they can come out whenever they are pressured by any sort of stressor. And we know about this with herpes, whether we're talking about cold sores or we're talking about genital herpes. Most of us know how that works. You can get the sores coming out at any time just from any sort of stressor of any kind, honestly, depending on who you are and what your immune system likes to do for fun. So they're not the only ones, though, that we would commonly think of. So there's other herpes viruses out there that you may not know are herpes viruses. Let me tell you 2 of them. Chickenpox and EBV, or Epstein-Barr virus, causes mono. They're both herpes viruses. So that makes sense now when we talk about that with chickenpox, yeah? Because it just comes back as shingles. You can have it for decades, and then you're old and your body's failing you. Say old, but they say it can come out as early as your 50s.
I had it.
Did you? For people that can, yeah. So that's a perfect example. So we know these viruses. We just didn't know that we knew them. But yeah, that's how they're working. So what time is it before I start going on with this? 6:40. I'll go through the questions. I'll read them to you. And if we have time for the end ones, then we'll do the end ones. I know you guys are probably not crying over it, but All right, what, because I didn't go through it, I didn't have time to go through it in my other classes. This is like the earliest I've been. Most of the time when I get to this point in my other classes, I have like 10 minutes left in the class. So we're doing pretty good, honestly. So viruses, why aren't they considered to be alive? Cells are alive. They can divide and make copies of themselves just with themselves. But these guys can't. They rely on the source of the host cell. So that's why we would consider them not to be alive. They're cheating. All right, what are the two essential components of a functional virion? The absolute two bare minimum, the nucleic acid genome, and the capsid, right? That's a bare minimum. What is the term for viruses that lack an envelope? I erased it up there, but that was naked, yes. What types of genomes can viruses have? That, all of that, that's possible. And then what is reverse transcriptase? It allows us to go from RNA back into DNA, which these viruses, some of them can use to put the DNA then into your own genome. All right, so let's move on to the life cycle of these guys. They all have the same steps, pretty much, no matter what. Adsorption, penetration, uncoding, synthesis, assembly, and release. And we're going to go through each step. If I say adsorption, I say it weird like that. It's adsorption, but if I say it that way, people think I'm saying absorption, and I'm not. It's a different thing. So adsorption is like sticking onto something. So we're going to dock into our cells. Okay. So we're going to come and adsorb onto whatever we were saying key. That's what I was looking over. I was looking up. I was like, where's I was talking about key, right? Yes, our spikes are going to be our key. They're going and finding the lock on the cell that they're coming up to, okay? Once they find a cell that has a correct lock that key fits into, then we can go on with the next step. But that step, just finding the lock, is adsorption, okay? Bound to our lock, we bound to the molecule on the surface of the cell, our host cell. This determines host range. So when we're talking about host range, we're talking about things like with the liver cells, right? Which I mentioned already. You can only infect human liver cells like hepatitis B does. It's very limited host range. That's the only things that you can infect. If we're talking about down at the bottom, broad host range, or rabies virus can infect cells of all mammals. So if you're a mammal, you are susceptible to rabies virus, end all be all. Okay. I had a student ask me in the last class, what about possums? Possums are not technically mammals, so they don't get rabies. Isn't that nice? they look like terror is happening, like in embodiment of terror. So they've got those teeth, man, and they'll just sit there and hiss at you, and then they'll pretend like they're dead. I get possums all the time in my yard. No. So you're saying I can have one? Yeah, you could. And so we keep finding baby possums, like when it's that time of year and there's baby possums, and the mamas will traipse through our yard, and then we have dogs, so the dogs scare the mama off, so she loses a baby. So keep finding them, and depending on how long they are, they can be released just into the wild at that point. So we found this out through our several times of dealing with these possums. I don't live in the wild, guys. I live in Bethany, so it's not like. But I live really close to the lake. I live like a street over from the lake, and so right by the Stinchcombe Wildlife Preserve. So I think that's why we have so many, but get a lot of raccoons too. They break into our sunroom. But anyways, they can't get rabies. We learned about that, and we learned about how, like, when they play dead, they're not, of course, they're not hurt at all. It's hard to tell sometimes. I would like poke at it and be like, "Are you okay?" It's like, my dog had you. I don't know. But anyways, let's talk about rabies just for a second, because this is the thing I was talking about. I love to tell people about with rabies. Okay, yes, it can infect mammals. And not possums, but yes to raccoons, yes to skunks, and yes to bats. Those are the most common reservoirs here in the United States for rabies. There's other ones as well, of course, but those are the common ones. And here in Oklahoma, I think skunks is number one for Oklahoma, but still, don't mess around with wildlife if you don't have to. If they are, out at nighttime and they're foraging for food and they're chilling and they don't want to be near you, they probably don't have rabies. Okay. If you see these videos on Instagram where there's like a mom, let's say the mom, okay, because I know there's like two different versions of this kind of situation. The mom who's like trying to get into the house and there's like a freaking raccoon that comes and attacks a mom. She like punts it across the yard and like runs into the house, right? Or there's the dad that's, you know, has the kid and he gets bitten by the raccoon. He drops the kid, runs. But whatever it is, if there's a raccoon attacking you in daylight when they're not usually supposed to be out in daylight, and it's attacking people where normally they don't want to be anything near you, it probably has rabies. Like, no joke, okay? So is that something that we should be concerned about? There's a reason why we vaccinate our pets and why it's required if you go to like most dog parks that you have actual rabies tags on your dog to prove that you have, vaccinated your dog. Or your cats should be vaccinated as well, especially if you ever think that the cat's ever going to get outside, be sure that it's vaccinated. Vaccine in general, guys, that's just in general. But with rabies, we become so accustomed to it here in America, relying on our pets having been vaccinated, that we forget what rabies is all about and why we vaccinate for it. So in those situations, let's say you are the mom and the raccoon comes to attack your kid, and let's say it bites your kid, okay? And yeah, and then you punch it across the yard and you bring out the shotgun and whatever. So, so your kid got bitten. Probably most of the people in here would be like, I'm taking my kid to the ER right away. Like that's going, like we're going to the ER or urgent care or something, right? And then the doctors there would tell you, okay, we're going to give your kid an immune globulin for rabies just to be safe. We're going to give them vaccines for rabies just to be safe. And you have to get several shots in series over this in order for it to be protective. The immune globulin is antibodies that are basically going to block the virus from getting into the cells. And the vaccine is there to help you make your own antibodies in the time being, because you can't just make them right up front. It takes a long time to get your, immunity going. And that's fine. And the doctors might even play it down some. They're like, oh yeah, we'll do this just to be safe and whatever, and blah, 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 blah. What would happen? What do you think the odds are if you took your kid and just decided to clean up the wound and wash it, soap and water, clean it really good, maybe some hydrogen peroxide or alcohol, and put, Neosporin on it and a Band-Aid, and just make sure that it's not getting infected. If your kid, let's say over a little period of time, I'll tell you how long in a minute, starts to develop any sign of rabies, whether that is a fever or muscle twitching, or it's not obvious at first, right? It's small stuff, and then you build up to the weird stuff. What do you think the odds are if you took your kid in then that they would be fine? They could be treated and be fine. Anybody know? It's none. It's a 100% mortality rate. That's why we vaccinate y'all. That's why we vaccinate our pets, because we don't want that anywhere near us. It's 100% mortality rate if you start showing symptoms. It could be potentially that way before you show symptoms, but for sure, if you get to symptom level, you will die. There's nothing they can do for you. And that is terrifying, and it should be terrifying. That's why we vaccinate, right? So keep that in mind now, that if you guys are going on in your lives, you can go terrorize other people with the story and tell them about how important it is to vaccinate pets, especially. But of course, don't let bats in your house. If there's skunks or something on your property and they're doing normal nighttime things, it might not be a big deal. But if you see them out and then doing abnormal things, I drove up here one time over by like the cops headquarters area, head pond. I normally come from the other side, but one day I went that way, and it was a nice sunny day, and there was a coyote, like just laying in the sun right by the road. And like, I was like, oh, this cute coyote. And I'm not thinking to do that. And then I get into like work, I'm like, this cute coyote. And then the people I'm working with, they're like, so maybe it's rabid, because they don't usually come out around people, especially in the daytime. It might be rabid, okay? But like, it was still cute, so. But yeah, so just be aware of this. This is definitely something you want to be educated on. I feel like, and I think you'll agree with me now at this point, that it's one of those things that is very understated and should be more, people should be more aware of it. So I'll talk about it probably more than that in this course, because it's one of my favorite things to talk about. But yeah, it's no joke. Like, there's videos, if you guys are interested, and I usually try to show them, but I might not get to it this semester, but there's videos of a man, like a grown man, that he allowed them to video him while he is dying from rabies. And you can watch it on YouTube. It's just on YouTube. But it has like an age restriction on it, of course. It's literally a man dying from rabies. But he goes through exactly what you would think. We know rabies as being, you've seen Old Yeller, for example, hydrophobia, right? A fear of water. They don't have a fear of water. They just can't swallow. So they will drown if they try to drink water. So they're not really afraid of the water itself, maybe a fear of drowning from the water they're trying to drink. That's because the throat muscles give out kind of early on in rabies. So they can't swallow. As rabies would have it, it likes to concentrate itself. It grows very well in the nerves, and that's how it moves into your brain and starts dealing with all these other symptoms. Yeah. But it also likes to congregate in the salivary glands. And so you cannot swallow your spit, you cannot swallow the saliva, and you start, if you're an animal, especially foaming at the mouth, and humans will do it as well because they can't swallow their own saliva. So you've got all this like rabies-ridden saliva in a mouth, plus they're triggered by the virus to be aggressive, to go attack whatever's near them, and all this. That's part of the neurological aspect of it. So it's just terrifyingly beautifully designed to work so well for that virus, being high concentration in the saliva, and you can't swallow it, and you're going to be aggressive, and then that's how you pass it on. And it's just a virus, I believe it's very simple. Yeah. Yeah, so that's why. So now you guys know, yeah. Now you guys know whenever they talk about zombie movies and they're like, oh, it came from, it developed from rabies or whatever, rabies virus. It makes sense actually a little bit.
Decided that pets were like the main transmitter of this, so that makes more sense to vaccinate them than it does to vaccinate us.
Well, yes. So in a way, they're more likely to get it than we are. And since they're the animals that we're around the most, we vaccinate them, and that protects us. Essentially, it's like a herd immunity concept, but on a different level. You can absolutely get vaccinated against rabies. If you're working in the vet industry, they'll probably vaccinate you, especially if you're like a permanent vestige. So proper, vet techs that are working somewhere, and proper veterinarians, obviously, if you're working at a place where you're going to have high traffic of dogs and cats, they'll probably vaccinate you. Yeah. Yeah. But yeah, it depends on your vet. Yeah. Yes. Yeah. It depends on the vet that you work for, of course, because they're not required to by law, but yeah, some of them will do it. I imagine it is. Yeah. I don't know. Yeah. Okay. Yeah. There you go. I bet it's not covered by insurance either because they're like, you were stupid and got bit by a dog. I hate insurance companies. They like send you letters in the mail being like, oh, you had to go to the ER when your leg was cut off. Why were you at the ER then? Would you really need to be there? Like, that's what I get letters from them. It's like that. So anyways, the penetration and uncoding of these viruses. We've docked on there. We have adsorbed. And now we're going to get inside of the cell. There's kind of two ways you can go. Once you dock, the cell can sort of envelop you in another envelope, but of just its own membrane, into a vesicle. Sort of like when we talk about phagocytosis, except this is triggered by like it thinks friendly mechanisms, right? So it's just taking you up normally. So it's called endocytosis. So endo into cytosis of the cell. Then in that vesicle, it can break down the parts of the virus, and that will eventually lead to freeing the DNA or RNA into the cell. The other way you can go is with your membrane here. It's a fancy membrane, but it's a membrane, okay? When you dock, you can like merge your blanket that you covered yourself with, you dock in, and then merge it back in to the membrane that you docked into. So it becomes part of that again. And then your nucleocapsid is ejected into the cell that way. Obviously, you have to have a membrane to do that one. But that's two ways that they can get into the cell. So that's the penetration and the uncoding. You get your nucleocapsid in there, how do you free your DNA? Typically, the cell that it just entered, your cells, are going to just do that for it. Break it down. Then it's going to replicate. It could be in the cytoplasm or the nucleus, because y'all, I told you that DNA only happens in the nucleus in our cells. So all the machinery and the parts and everything for making DNA is in the nucleus. So if you're a virus and you are DNA virus, then you have to go into the nucleus to do all this. The RNA guys can just do this out in the cytoplasm. I say cytoplasm, the ribosomes, they don't have to always be on the replicar. They can just be chilling in the cytoplasm. So they can be doing that out there. But this is a good picture that depicts kind of how it would happen. You have your adsorption in step number 1, which is up at the very top with our spikes. Then we will penetrate and uncoat as the cell takes up that cell. So here's the uncoating here. We're freeing our RNA in this case, and it's positive sense. So we can just start reading it and start making protein if we want to. So here we can get capsomeres and spikes. The spikes are going to go on the surface of the cell. So the capsomeres are going to start forming around copies that we've made of the positive sense RNA. If you're a positive sense RNA and that's your genome, you have to make a negative sense to make more positive sense. Okay, so it's just how it goes. It's like you have to have both sides of the strands going if you're going to do all this at some point. Okay, but yeah, so we got our positive sense RNAs that can actually be, encapsulated, if you will, by those capsids. And they'll get their capsid around them. They'll become the nucleocapsid, which is what this is, nucleocapsid. And it's going to bud out and surround itself with that membrane of your cells that already has the spike proteins on it. It's called budding. It pushes its way through and gets released that way. What if, though, you don't have a membrane? How do they get out? Because they don't bud out, right? They don't need the membrane on the outside. How they get out is the cell can just called exocytosis. The cell can just spit you out. It's like you go through the whole Golgi apparatus, the packaging and transport organelle. You can go through that and then just have it transport you out of the cell. So you just like it would any other toxin or signaling molecule or anything. That's exocytosis. So that can happen if you don't have an envelope. This is just depicting how it works with the DNA. Guys, they have to do all the DNA replication in the nucleus. So they'll have to bring their mRNA out in order to be translated and then bring their translated proteins back in to build the viruses. And then those can actually bud out of that nuclear membrane. They don't need to bud out of that main membrane. Here's a depiction of a cell. This whole thing is one big old cell. This whole thing here. And then this thing in the middle, this structure here, is a nucleus. And inside of that nucleus, we can see viral particles here. That's all these dots. All viruses. They're just adenovirus. Very tame. And then this is very tame cold-causing virus. In fact, you might not even get symptoms from it. This is just a buildup of the proteins that are used to make the capsids, the capsomeres, and then the capsids. That's all that is. It's just buildup of protein. Look at how messed up that cell is now. Because like we usually see like a little bit of nucleus and then all these structures around it. This has been completely taken over in the nucleus here. So that's a hot mess. So once you have your mature viruses, we already went through this, the budding or the exocytosis, I say endocytosis. Exocytosis to push something out. Endocytosis taking it in, exocytosis to push it out. That's the two ways you can do that. And then we talk about the damages that are done to the host cell. These are called cytopathic effects. Cyto is cell, pathic is disease, and then effects. Okay, so things that are done to the cell as a result of being infected. This could be including looking like we did in that other picture, microscopic appearance of the cell, that massive nucleus, right? The cells can become disoriented. They don't know like what's up and down to them. Like usually you might have an up that's binding with one part of a cell, but now you're not doing what you're supposed to do. They can undergo major changes in shape, size, and intracellular damage. Inclusion bodies are just areas where we have buildup of virus or damaged organelles, kind of like we saw in that nucleus picture, right? That's an inclusion body of all the proteins. And then we have syncytia. Syncytia is where we have a fusion of multiple host cells into large cells, because I'm telling you that on the surface of those cells, we have those spike proteins, yeah? And those are the keys to get into the locks on cells. And now we have them on our cells, waiting for viruses to bud out. But in the meantime, they can be keys trying to get into the lock in the cell next door. So what happens is they do that, and then that merges those cells together. You can have many cells merged together that way to form, one giant cell. So that's what syncytia are. If you've ever heard of RSV, respiratory syncytial virus. So they do this as well. So that's something that can go bad with your cells. There's another one that's not on here, but it can affect adherence of the cells to whatever, don't know what to call it, like other cells or like the extracellular matrix or whatever it is. They're not adhering like they should. You imagine that you have cells that are in your red blood vessels, your endothelial cells in your red blood vessels that are not, adhering like they should to the cells that are near them. That can make your blood vessels very fragile and break very easily. And that is where we get hemorrhagic fevers from. Because eventually those blood vessels become so fragile that they're just not holding together. And you can literally just start bleeding out without any effort at all. It starts off small, yeah. That's where you get Ebola and stuff like that. Just from viruses, y'all. Okay, so so we have the proviruses. I can see why I always liked viruses. We're almost done. So the provirus is you insert the DNA of HIV or HSV, herpes simplex, and put it in the DNA of the host, and that is the provirus. Then once it's in there, okay, it can still make virus from that, but it usually will have to be activated somehow, like we do with herpes. So that's a provirus. That's when we're talking about normal viruses in eukaryotic cells. You can imagine then that you can have a provirus sticking into places that are damaging to how the DNA functions. So what I mean is your cell cycle and mitosis or something like that gets interrupted because of how these proviruses insert themselves. Those are oncogenic viruses that tend to do that. And that process is called transformation, transforming those cells into cancer cells. So that's what happens with HPV. We estimate that about up to 13% of all known human cancers are actually caused by viruses. We estimate it because we don't know what viruses are causing them. We just think that they're probably caused by viruses. That's kind of scary to think of. All right, bacteriophages, don't really care about the history of them. These are viruses that are going to be infecting bacteria, and I'm not going to ask you about their genome. We already talked about the T4, and it's basically going to take this crazy little body on its feet. The feet are the spikes, okay, the long legs. They come and recognize that it's E. coli. And then when it recognizes E. coli, it squats down, puts its little tail fibers, sticks them into the wall of the bacterial cell, and then causes that coil to sort of spring down, and that injects the nucleic acid into the cell. Isn't that nuts? And so it does that, and that's what they look like when they do it. And so it's injecting its nucleic acid in. It doesn't need to go in. It doesn't go in. In fact, no bacteriophages go into bacteria. They only ever inject their genome in. Then the bacteria are going to make copies of that bacteriophage using just the genome. And then they are just going to burst out. They don't bud out. They don't get exocytosed out. They're just going to make so many virus particles, the phage particles, that they're just going to burst the cell open. Okay. Here's a picture of that happening. Is this cell getting infected with these phages? Isn't it so neat looking? There's two cycles that you can go through if you are some of the bacteriophages can do this. Not all of them can, but some of them can go through two cycles. The lytic cycle and the lysogenic cycle. The way I remember which one is which, the lysogenic cycle is going to involve genes. I'll tell you what that means, but the lytic cycle is the normal one that we've been talking about this whole time. Making the viruses and spitting them out, okay? The lysogenic cycle, though, is where, for bacteria, they're incorporating the virus into their cells. So I know we've called it the provirus for our cells. For them, it's the prophage. The prophage is incorporated into the bacterial genome. That's the lysogenic state. Like I said, genes is how I remember that one, the GE and the gen part. Okay. So when the genes are part of the genes, that's the lysogenic state. The lytic is the normal state, right? And they'll just wait to get turned back on again. There's usually an induction event that will induce them to start making more viruses again. The phages that can do this are called temperate phages, but they can switch back and forth. I already told you about all this, yes. Lysogenic conversion is something that can happen where if you are a bacteria and you get infected by a phage that's carrying a special gene, its little DNA, and that special gene and its DNA get incorporated into your DNA in lysogeny, that lysogenic state. Okay? That can give you toxins, like the toxin that is responsible for causing diphtheria when you have diphtheria, the toxin that's associated with cholera. There's actually a couple of toxins associated with cholera that we can attribute to phages actually infecting these bacteria and giving them these toxins. Botulism is another one. So they've actually gotten their toxins from phage infections, which is kind of crazy about, but that's how they get them. If you don't know this, the Tdap vaccine, the D in the Tdap is going to be diphtheria. And that toxin is what you're actually vaccinating against. It's not the bacteria. And it's the same thing with tetanus. You're actually vaccinating against the toxin. And the same thing with pertussis. You're vaccinating against the toxin. So your body can get rid of that toxin so it doesn't cause you negative effects. Your body doesn't care if the bacteria get in there after that, because who cares about the bacteria? It doesn't care. They don't do anything wrong to you if it doesn't have the toxin. Really weird to think about, but
Is that why those can go together in one vaccine?
yes, they kind of have to. And we'll talk about that when we get into immunology later, but that is a very good point. Very good observation. When you need to grow viruses for studying them, you have to have cells to grow them in. That's really all this is talking about. It's going to be a pain. And we know that there are impossible to measure the number of viral infections worldwide. We know there's viruses all over the world causing all sorts of different infections. Some of them are debilitating, some of them are deadly, and some of them are mild. There's something, this is the last bit. Oh no, it isn't, because there's the prions. Okay, we'll do this and then the prions, okay? The interferon. If you are in your house and somebody's breaking into your house, right? And he's like going around shooting the people in your house or whatever, and you're in your closet and you have your cell phone, right? And you call your neighbor and you're like, Jimmy, we got a situation. You probably want to get ready. I'm probably not gonna make it, but you probably want to get ready. So Jimmy's your next door neighbor. So Jimmy gets out his shotgun, and everybody in their family has their shotguns, and they're all waiting at the door for this robber, right? That's interferon, is that phone call. So interferon tells the next cell that there's a viral infection going on, and it lets them know so they can start making antiviral proteins and getting ready for that ahead of time. So really cool thing that your body can do. It's also used in a lot of different treatments. All right, last things, I promise, is the prions. Prions are misfolded proteins. We call them infectious proteinaceous particles. They're literally just misfolded proteins. It's a normal protein that's in everybody's brain, and most mammals have this protein, but it just misfolds. We don't know why. We don't know what causes it, but it just does. And it doesn't seem to be genetic either. So it can be, it's heritable, but we can't find genes for this. So we don't know what's going on with that. But we don't fully understand it. Misfolded proteins, you can get it from being exposed to other people's misfolded proteins, or cows' misfolded proteins. We're talking about mad cow disease. So you go back into like the 90s, and you eat a burger in Britain, and then you go and chill in America after your nice trip in Britain, and 30 years later, you start having symptoms of mad cow disease. And it develops the way that people would get it, but it's pretty similar. It develops like dementia at first. It's a lot like Alzheimer's or dementia, and then eventually you'll die from it. It's 100% fatal because it's just a misfolded protein. It doesn't have a genome. What do you do? It's a normal protein. You can't make an antibody against it either. So it's just nuts. But yeah, so these guys are folded in a way that's super resilient. You can't autoclave them. They'll even resist like certain temperatures of incineration. You have to get certain temperature in order to burn them away. It's like they are the optimal folding of this protein. It's not nice folding for your body to function, but it's the optimal folding for them to be stable. It's just insane. So that's why even if you cooked your meat, it wouldn't matter. It's still gonna be there. You could like practically char it. So, anyways, so this protein will get into the nervous system, and because of the way it's misfolded, we know about our hydrophilic and our hydrophobic, yeah, and how proteins want to fold, and we don't want the hydrophobic, the water fearing, on the outside. It doesn't do well with the rest of the cell stuff. We usually hide them on the inside of our proteins. These guys misfold and do it differently. They have it on the outside, but that causes them to stick together. So they're trying to hide from it by sticking together. That's what the buildup that is associated with that is what causes holes in the brain, why we call it spongy form. So spongy, like sponges, the spongy form. And it's an encephalopathy. Encephalo means brain, and pathy means disease. It's not a crazy word, it's not as crazy as it looks. So that's what that is. Mad cow disease is one version. You can have it just naturally in people. It's called Creutzfeldt-Jakob disease. It can be heritable. We can see it in families. We can also see it as in different formats. Like we can see it as fatal familial insomnia, where it seems to be heritable there as well, but people die from not sleeping. Literally, like the protein just makes it so that their brains like don't work in a way they cannot sleep, and eventually they die from the not sleeping, not from the brain damage, from the not sleeping, because they just can't. They're physically incapable of it. I can't imagine the frustration involved in that. But you die from that, and it's super rare, of course. And then there's one called Kuru. If you haven't heard Kuru, it is a version of this prion disease that is going to happen in Papua New Guinea, I believe, tribes that they venerate their dead by eating them. So yeah, and they're not hunting people or anything, but they're just venerating their dead. And they think it's a lovely practice to incorporate them as part of their body and all that. But they are just continuing to spread this prion disease in their tribes by eating these people. So that's called kuru. But yeah, there's other animal versions called scrape beef for cows and all this. So we don't know exactly how it starts, but we know how it gets transmitted after that. There's no nucleic acid, there's no genome, nothing. It's just a misfolded protein. And that's that. I know it took a long time. That's it. So we're going to go up to the lab now. Or anybody have any questions, I should say. We can go to the lab.